Hello and a warm welcome to today's ISNTD Connect, our weekly meeting on all things neglected tropical diseases. I'm Marianne Compret and I'm speaking on behalf of the International Society for Neglected Tropical Diseases. And it's my great pleasure to welcome you today for this session in collaboration with the Global Schistosomiasis Alliance, where we will be taking a closer look at some really exciting innovation in schistosomiasis diagnostics. As many, if not all on this call will know, schistosomiasis is one of the most prevalent neglected tropical diseases. And despite some important progress in its control, as well as the well-established diagnostic methods and tools currently in use, innovation in this field will be hugely important to improving and refining the understanding of the true prevalence, but also the response needed to this disease, particularly as this very prevalence decreases and elimination targets come closer and often in the face of very limited resources. So we're delighted today to welcome our three speakers who will take us through some very promising results, as well as really an exciting vision of what the future of schistosomiasis diagnostics is likely to look like. So please join me in welcoming very warmly, firstly, Dr. Bonnie Webster. Hi, Bonnie.
Hi.
Hello, welcome. You're principal researcher at the hugely active Life Sciences Department at the Natural History Museum in London. Thanks for joining us. We will also hear from Dr. Andy Wende, founder and managing director at Xpedite Diagnostics. Hello, Andy. So, a real powerhouse in R&D and invention in point-of-care diagnostics. Thank you for joining us. And we'll also hear, following those two presentations, from Dr. Michael Frimpong. Nice to see you, Michael. Hello. And Michael, you are currently a fellow at the National Institute of Allergy and Infectious Diseases in the USA. You are also representing the Kwame Nkrumah University of Science and Technology, Ghana. You're a grantee of the African Academy of Science, and it's really a huge honor and pleasure to have you with us today on this call.
Thank you.
So thank you to all our speakers for joining us. A huge thank you as well for all the attendees who are joining the call one by one. Really lovely to see you here. I can see this is going to be a really dynamic and exciting session. The chat is already, without any prompt from my part, just really active. Everyone's saying hello. Welcome, Inge Kreutel. Nice to have you here. Birgit Jindig-Venevald, hello, Solomon Wafula, and everyone else, please don't hesitate. Join the chat, tell us who you are, where you're tuning in from, which organization, which country, and of course, as and when any questions arise during the presentations, please post them there and we will address those after the presentations when we get to the discussion part. So without any further delay, I'm going to hand over to Bonnie. I thank you all for being here today.
Thank you very much, and thank you for that introduction. And thanks to the audience for joining us today. So many of you in the audience be fully aware that we need better diagnostics if we're going to reach and maintain elimination of schistosomiasis. And this is really accentuated by the complex life cycle of schistosomes, where a single infected person can infect a population of snails, which can then go on to produce many thousands of cercariae, facilitated by this clonal replication. And so resurgence of transmission can happen very easily and very quickly. However, developing new or optimizing existing diagnostics of schistosomiasis is not easy, and this is mainly due to the diversity of the disease in terms of the different endemic settings that we see, as you can see here in these images, the different populations at risk, and the different disease pathologies that we encounter as well. But also it's complicated in where these diagnostics are needed and where they need to be implemented. For example, are they needed at the point of care in environments like schools? Are they needed in environments such as these rural or community settings? Or are we going to try and implement them in basic clinical settings or healthcare facilities? So one of the research focuses, and particularly what I'm going to talk today in our lab, is the development of molecular assays for many aspects of schistosomiasis. and I'm going to go through how we develop those assays over time. But we also know that assays developed in labs can work extremely well. And, but there's, hold on, it's just not moving, there we go. But there is also a big gap that needs to overcome to actually get them to work properly in endemic settings. So you can see this bit of funky artwork, which I put together for this talk, and you can see a very happy scientist in their lab, and they've developed an assay where it's worked very, very well. But then when we start to think about the endemic settings that we need to implement in, it becomes much more complex. There's lots of variables that we encounter, and there's lots of questions that arise to actually overcome this gap. We can start to put some of the pieces of the puzzle together to actually bridge this gap, and sometimes we can reach the other side. But even when we reach the other side, many hurdles may be put in our path to actually get these diagnostics feasible and implementable at a wide geographical scale. It's also very important to work with others along this journey, and particularly I'd just like to highlight partnerships between industry as well to actually bridge some of these gaps. And this really allows us to advance these diagnostics forward. So just to really kickstart my talk, I just want to really acknowledge the people that I've worked with along our journey for molecular diagnostics. There's a whole team at the Naturalist Museum, and particularly we work with a lot of students that developed these diagnostics throughout their master's projects. I work very closely with the Leiden University Medical Center, as they're real experts in PCR-based diagnostics. And then out in the field, we've done a lot of work out in Zanzibar, with Steffi Knopp and Charlie Aim. I'm also going to talk a bit today about female genital schistosomiasis. I work very closely with Amaya and also the team out in Zambart, and I'm working out how we can develop better diagnostics for female genital schistosomiasis. And two real key partnerships that have helped the journey for this diagnostics is working with Long-Haul Diagnostics in America, but also Andy Wende and Ludovic at Xpedite, who are presenting today. So ideal diagnostic characteristics for diagnostic schistosomiasis really does follow this reassured criteria, and we need to try and fit our diagnostics into these criteria if they're going to be feasible and implementable at the point in endemic settings. But some of these characteristics are quite difficult to rise to. We know we need high sensitivity, they need to be affordable, they need to be simple, they need to be quick, robust, they need to need a little equipment as possible, and they need to be a deliverable. And when we think about deliverable, it's all about access, availability, affordability, acceptability, and also they need to be workable and used in an appropriate way. And there's a direct link between accuracy of the diagnostic and cost and simplicity. As we know, if a diagnostic needs to be highly accurate, it is often more costly, and it's also not so simple to run. So as we move towards down the roadmap for schistosomiasis elimination, and we're trying to reach the goals set out by the WHO, we know that we have severe gaps in the diagnostics that we need, not just to reach elimination, but also to maintain elimination. And now has been published the target product profile for diagnostics for schistosomiasis, and this really sets out the ideal diagnostic characteristics needed. And this is now split into two categories: diagnostics for monitoring and evaluation, and diagnostics for transmission, interruption, surveillance. But we also know as we move through a control program, moving as intensity and prevalence starts to decrease, our focus on control changes, and so does our diagnostic needs. And the diagnostics we're going to use along the way will change. We move from morbidity to infection and transmission control, down towards elimination, and then also down to surveillance. And this also relates to our treatment interventions that we're going to implement, moving from MDA to selective treatment and down to test and treat. And as we move down this path, our diagnostics need to become much more accurate. So we're really focused on this middle area of the control program, where we need really high sensitive and specific diagnostics to actually diagnose low levels of infection and intensity of infections. And there's two main forms which are of keen interest here. There's the antigen test, the CAA, and the CAA, which is the more recent one coming through, and also the molecular tests. And I'm talking about the molecular tests today. So the molecular tests can be split into two main categories: PCR, which often needs quite supported laboratories, and the isothermal assays, which I'm going to be presenting, and also Andy and Michael will be presenting about these isothermal assays as well. And the reason I work on molecular assays is really because they're multi-purpose. They're not only useful for diagnosing infections in humans and animals in the clinical setting, they're also very useful, and you can start to develop them for other aspects, and you can use the same platforms for various aspects of schistosomiasis transmission monitoring. We can use them to detect contamination of water samples. We can use them to detect environmental DNA, so we can actually detect transmission and risk, and show where risk of infection is, and snail xenom monitoring as well. So it's very useful to have use molecular platforms and use them to develop for multiple areas of detecting schistosomiasis transmission. So our work is particularly focused on isothermal assays, and this table at the top just puts the different characteristics of 3 molecular assays. There's the qPCR assays, which is the assay used for most reference testing. There's LAMP, which is the most commonly known isothermal assay, and there's RPA, which which a lot of our work has been focusing on. RPA is also known as RAA, which is recombinase aided amplification. And you can see the different characteristics of these assays. You can see that for RPA, it's a very quick and low temperature assay, and it can be quite simple, and it can be portable. So an isothermal assay is an assay that runs at a constant temperature, and we don't have to increase or decrease that temperature over time. It still follows the same principles as any molecular assay, where we are looking for or taking out DNA from a sample. We need to amplify it so we can detect it. And then we need to have some form of way of detecting that DNA amplification. An RPA can use multiple ways of detecting that DNA amplification, from fluorescence to small portable readers, and also these DNA dipsticks. But I also just want to focus, so we're not completely set on using RPA going forward. We also explore other assays. We're also very interested in LAMP assays, particularly as those platforms are much more common and are advancing in their technology very, very fast. And we're also interested in these portable qPCR systems. There's some very good mini qPCR systems coming available, and several of these have also been tested in endemic settings. But to focus on the RPA work today, this just shows you a workflow that we've been developing for urogenital schistosomiasis, detecting schistosoma haematobium eggs, and also, we hope, cell-free parasite DNA in urine samples. So this work has been ongoing for several years now, and we've got to this optimized workflow where we take a sample, we do some sort of extraction. I'll go into more detail about that. We take the DNA out of the sample, either in a crude way or in a pure way. We have our assay, which we can set up by adding buffers. The reagents come in a lyophilized pellet, and then we use small portable fluorometers to detect amplification and give us a positive diagnosis. We've also adapted this to female genital schistosomiasis, and I'll show you some of the work on that. And we have pilot data to show that we can detect infections in snails as well. At the end of my talk today, I'll also talk about some of our work that we've been progressing on Schistosoma mannsoni, as that is the next assay that we'd really like to focus on. But again, working with stool sample is problematic, and that's why for our work we actually focused on urine when we first started. Stool samples contain many inhibitors, and they're also very difficult to work with. So the pilot work we did on our haematobium assay, which is focused on a repeat region, a DNA repeat region known as trial 1, which is a commonly used DNA region, really gave us great results. This is this is all very much laboratory-based work where we have very good facilities. But what we did show is that we can detect down to a single egg spiked in the urine sample. So it showed us that our limit detection was low enough to detect single eggs. We then were able to take this and test it in a pilot study on clinical samples, and we're very fortunate to be able to partner with the ZEST study, the Zanzibar Elimination Study, run by a school. And within this study, there's a biobank of frozen urine samples which are ready available for diagnostic testing and development. So within this study, we had to pick a sample preparation method. And through literature researches, we were able to come across a very simple and crude preparation method, which we just had to pilot test out in Zanzibar. So this test just then brings us to partnership with Xpedite Diagnostics, Andy Wende, and it was actually his protocol that we all used. At the time, it was known as speed extract, so you'll see that in the literature. Now it's known as Swift DNA. So we're able to use this protocol to do a crude extraction. And you'll go into more detail about this, but what it does, it allows you to use a magnetic rack, which is portable, small heater, and then using magnetic beach, you can very quickly isolate DNA from clinical samples. And the data from Zanzibar was very promising. The samples that we're using were quite crude. We were just taking a random aliquot from urine samples. Many of these had been pre-filtered to look for eggs in 10 ml samples. So we weren't doing any proper work on how we were actually dealing with that sample at the time. We were very interested in these samples that had very low egg counts, particularly between 1 to 10 eggs per 10 mils of filtered urine, as that's the kind of area that we need to focus on in terms of accurate and sensitive diagnostics for elimination. So our sensitivity was between at the low 90s for the assay then. But just to show you the kind of data that comes out of these urine samples in this very low egg count, you can see here these are egg count data for a selection of the urine samples that were tested. You can see for microscopy, we get discrepancies in our egg counts, which show that the egg detection is very difficult, so they're really low intensity infections. And again, we were using random aliquots of samples that are left over after the 10 ml filtration had been used. But again, it gave us very promising data to move forward with. But following on for this work, we really wanted to focus back on that sample preparation stage. This was the main hurdle in moving forward with more field implementation, particularly this heating step here. Portable heaters are available, but you do need some form of electricity to power them, and also they can take some time to heat up as well. So we really wanted to simplify this sample preparation stage. So in the laboratory, we went back, and this is again a master's project led by Owen Donnelly and also supported by Silver Mosquito in our lab to try and simplify this protocol. And what we did, we could, using samples within our lab, these are frozen eggs taken from laboratory rodents, we were able to reduce the steps needed in the sample preparation. We could remove the heating step, and we could get very good results from single eggs using this method as well. So that showed us that we could actually simplify our sample preparation, which gives us more feasibility for implementation in endemic settings. Owen also did a lot of work on his project looking at different sample preparations and how that affects our RPA assay. And this work's really important because it really shows you how you need to complement, you have to work with your molecular assay and also the extraction method, and they need to fit each other because one can affect another, particularly certain extraction methods. So what Owen did for his project is he took different types of extraction method. This one at the top is a simple lysis, and we use different temperatures. The one in the middle is a Quiogen kit, and Quiogen is a very common laboratory commercial supplier. And again, we use different temperatures using their standard kits. We used a kit from Primer Design, which was actually designed for field use, and we used other methods as well and also putting eggs directly into our assay. And you can see we get different results. You can see Quiogen actually, the enzymes in the Quiogen kits actually affect the RPA assay, and it does something to the enzymes in the reagents. So again, this shows that it's probably not very good going forward. but you can see the primer design completely failed again. So it's very important to look at different extraction methods and couple it with your assay as well. So the really next steps for this work is to really focus on a lot on that sample preparation stage. We need to figure out, particularly with these low-level infections, low-intensity infections where we have very few eggs, we need to figure out if we can just take a random sample. Is that going to be sensitive enough? Do we need to concentrate the sample, or do we need to filter it as well? We're very fortunate to have some funding going forward, and I'll show some more details about that project at the end of my talk, from the Task Force for Global Health, and we'll be focusing on some of these key characteristics and also trying to tackle some of the other TP characteristics that are needed for diagnostics, such as that extraction method as well. So we're going to be testing different extraction methods and seeing how that affects our sensitivity and specificity. So I'm now going to move on to talk about FGS and show you how we've adapted our RPA assay for urogenital schistosomiasis onto FGS samples. So FGS is a chronic condition associated with urogenital schistosomiasis. It's very hard to diagnose. The main form of diagnosis is colposcopy, taking images and looking at lesions. This can be difficult. It can be, it's not always acceptable. And also some of the, there's some difficulties in diagnosis as it can be non-specific. You can take biopsies. This is also not very nice to do, increases risk of HIV. You can do symptom questionnaires, but again, this is not so specific. And there's also not a very good correlation between urine egg microscopy and FGS positivity as well. So it's very hard to diagnose. Amaya has done a lot of work on this, and working with the Leiden group of Grain has showed that you can use PCR methods on self-swab samples, really to show that you can pick up DNA, haematobium DNA in these self-swab samples. And this is a paper that focused on using quantitative PCR in that method. We took this study forward, and we were able to use some of these samples to actually show that to test our RPA assay on. We worked with both lavage samples and vaginal swab samples. We tested various extraction methods, and then we ran our RPA platform on those samples. And this work was led by John Archer. And again, we got nice promising results. We showed this is for lavage samples. We know that we can do very crude preparations on lavage samples, and we are getting a sensitivity of about 85% and a high sense of specificity. More excitingly, that when we use self-swab samples, the sensitivity of our assays went up into the 90s, showing that this had a very promising, it gave us very promising data to take this forward. Working with swabs is also much easier. We don't need to do any aliquoting. We don't need to do any concentration. So it's also a very nice sample type to work with. The platform was also really portable. This work was done during lockdown and COVID by John, and John was able to take our small laboratory back to his apartment, and he ran most of these samples in isolation within a small room in his apartment. We still don't know what kind of DNA we're detecting in these samples. We don't know if it's egg DNA, if it's cell-free DNA. And also we don't know about, are we picking up chronic or active FGS infections? We also wanted to try and simplify our sample preparation, and we had some good ideas about doing some simple bead-based extractions on swabs going forward. And what this led on to was actually to be involved in another FGS study led by Ginny Bond for the London School, but also based out in Zambia in Zambia. This was a social science project, but what we were allowed to do as part of this project is to start to allow women to produce self-swab samples to try and provide a diagnosis for FGS. So within this study, several hundred of women provided self-swab samples, and we were able to actually implement them and use them in our RPA platform. The hope within this study was that we would set up a portable lab out in Zambia, and we would implement the RPA platform out there. And again, as with many projects, COVID came and that disrupted all of those great plans. We also needed a very quick way, as all the lockdown started to happen, to allow women to preserve their swabs very quickly and to make them safe as well. This is where we had partnership with Longhorn Diagnostics, and what they allowed us to do is to use a transport media, which was very untested for these swab samples, to actually preserve those samples. At the time, we had no idea how that was going to impact our RPA assay, but we just had some discussions, and we made some plans, and we thought that we'd just give it a go and hopefully it would work. This transport meter is very useful because it preserves the sample, it allows it to be transported, and it also makes it non-infectious, particularly when you're working in HIV areas. This is quite important. But what we did do and have to do is we had to come up with a way of actually extracting the DNA from the sample. And working with Longhorn, we were able to develop this 4-step protocol, which is very simple and quick to take the swab and take the DNA from the swab. This is some pilot data that we got in our lab. And you can see we were spiking lung eggs on swabs, extracting the DNA. And these are the curves, the RPA curves we've been getting. This image at the top here just also shows you that we can shine blue light onto our assays, and we get fluorescence, which we can see using the naked eye. So again, this showed us that it worked and that we could use it to screen the samples out in Zambia. So we developed a platform. We developed a standard operating workflow, sample taken, sample preparation, doing the RPA assay, and then data interpretation. And we're very lucky to be able to work with Jennifer Fitzpatrick, who was going to be based out in Zambia. She came to our lab, we trained her, and we set her up with a small portable lab, which she took on the plane with her. And then she went and implemented the RPA out in the Zambia lab. So within the study, we were able to screen about 524 cell swabs. There was also small volume urine microscopy associated with these swabs. And this is the data that came through. It's a very low prevalence setting. So by RPA, we had FGS prevalence about 15.2%. Urine microscopy gave us 9.2% prevalence. And you can see we had concurrent data of about within 41 of our samples. Again, just to point out, it's a very low prevalence setting. So again, this is showing that the assay is sensitive. We need to do qPCR as a reference test to actually prove or compare the sensitivity and specificity. But it did show us that the RPA was feasible and it was transportable. We could do knowledge exchange to these labs. Again, we don't know if we are picking up active or chronic FGS. There is discrepancy between eggs and urine FGS, and we know that this is common. We don't know about treatment of FGS, but we do know that providing a diagnosis helps with stigma, and it also allows increasing awareness about FGS. Okay, so just to finish off, just a few slides now. For the first one, I just want to talk a little bit about work on Schistosoma mannsoni. As I said, working on Schistosoma mannsoni and stool samples is not easy. But this work was really kick-started by Silvia Mesquita, and it's related to schistosomiasis in Brazil, which is all intestinal schistosomiasis. Silvia Mesquita came to as a student placement, PhD student placement in my lab, and she was been working on diagnostics in Brazil throughout her PhD. It's a very low prevalence setting in Brazil. It's about the current prevalence about 5% with only 1% heavy infections, and this. The goals for elimination in Brazil is put through by the WHO. So Silvia developed a mannsoni-specific RPA assay in our lab known as SM-MIT RPA. She proved that it was highly specific and that it was highly sensitive. And she was also able to prove that we could pick up infections in a very small samples, small number of stool samples that we had, and also we could pick up infections in the snails. The sample preparation for these types of sampling is also needs quite heavy requirements. Again, that's something that we need to tackle going forward. Another exciting project that we've got ongoing on Schistosoma mannsoni is led by John Archer again, based at the Naturalist Museum, but also at Liverpool School. And John is doing a PhD supervised by Rossa Stathard. And he's also going to be looking at large numbers of fecal samples, looking at how we can extract DNA in crude ways and compare it to pure ways, and then also comparing qPCR, RPA, and LAMP as well. And then looking at costs of these assays and comparing them to see which one performs best and which just gives us the best data output, and also trying to implement them out in Malawi. And just to finish off, I'll just talk very briefly about our new project, MIDS, Molecular Isothermal Diagnostic Schistosomiasis, which we've got ongoing and will run throughout this year. So this is funded by the task force, as I mentioned earlier. And what this project really is focused on is bridging the gap between the lab and implementation in endemic settings. And what we're doing, we're taking our platform that we've developed, and we're really trying to fill those gaps for those characteristics that need to be filled so we can actually implement them out. And we'll be working out in Zanzibar again, in that really low prevalence setting to really test this diagnostic going forward. We're doing a refinement of the assay itself. We're looking at robustness and simplicity. This is just a very quick to show you the kind of thing we're doing. This is our snail room at the Natural Museum, where we're putting the reagents in tropical and hot temperatures and seeing how robust they are over time. We're looking at the speed and throughput of the assay and what bits of equipment we can start to remove from the platform. And also we're very much focused on the sample preparation stage, looking at how we're going to prepare the urine and how we're going to get the DNA out of it. And then the second stage of this project is really about field implementation. We're going to be doing quality assessment exercises with this both Swiss TPH and also out in Zanzibar. We're going to do transferability. We're going to try and do field implementation. And we're also going to do capacity building and knowledge exchange. We're going to also see how acceptable this diagnostic is, and also do full cost evaluations as well. And it's really about ambitious dreams. So at the end, we'd really like to have a very simple assay that works very well in these endemic settings, so that we actually can start to provide these really high sensitive and specific diagnostics going forward. And I'm going to finish there, and I'll hand back to Marianne.
Thank you so much, Bonnie. You speak of ambitious dreams, but I think we can all take a moment of silence and just really contemplate and consider how far you're turning those dreams into reality. So a hugely impressive body and amount of work, and it's really striking how right from the start you're addressing every single letter in the reassured kind of framework. So thank you so much for taking the time to share that with us. And what's been interesting is the huge amount of interest that is in our audience, interest from all around the world. We've got colleagues tuning in, many from Nigeria. So hello, Nigeria, and thank you for joining us. Celestin Madugos at the University of Kinshasa School of Medicine in DRC, all the way to Gunawan, Selamat Malam, tuning in from Indonesia, Claribel Murillo Solano from Colombia, Valbona Johnny in Albania, many colleagues in Germany as well, but also not only colleagues working in diagnostics or NTD, but Collins Onditi is tuning in from Kenya, working in safe water and AIDS, which of course are huge connections, whether it be with FGS or just the safe water in and around schistosomiasis. So really interesting work, and thank you so much for that, Bonnie. And without any further delay, I'm going to hand over to Andy Wende, who will take us through a bit more of the technical side of this work. Andy, over to you.
Yeah, so good afternoon, everyone. I'm going to talk about, let's say, field deployable tools for molecular diagnostics in low resource settings. Bonnie's talk greatly already showed also the value that these field deployable tools may bring into the whole schistosomiasis elimination and diagnostics in general field. So I'm starting very general, very basic, and then gradually going into more deeper realms of the technology that we can offer as Xpedite Diagnostics. So the basic principles of any molecular diagnostics is basically three main pillars. You have to sample your specimen. That means, yeah, sample collection, but also sample preservation if you don't process the sample directly. Second step is the DNA and RNA extraction itself. That means you have to disrupt your cells, whatever it is. It could be parasites, could be bacteria, or human cells. Then after disruption, you have to concentrate and purify the released nucleic acids. And the third step is the actual assay, where you look using different methodologies for the presence and maybe the quantity of the nucleic acid that you're looking for. That can be done using quantitative PCR, sometimes digital PCR, or as Bonnie has mentioned several times, also via different isothermal amplification methods. And recently it's also more and more coming that sequencing methods are also used for that. And the main expertise of Xpedite Diagnostics is lying in the middle part here, the DNA and RNA extraction. And that is why all these further slides will focus on that part. So what are the essential steps in the nucleic acid extraction itself? I mentioned it, it's the lysis, the disruption of cells to first release the nucleic acids. Then you have to make sure that nucleases present in the samples are inactivated to basically preserve the levels of DNA and RNA that you have in your specimen. And the next step is then a purification, which means you have to remove everything else that is not a nucleic acid. It can be proteins, lipids, carbohydrates, or secondary metabolites. And finally, usual nucleic acid purification methods also concentrate the DNA in the end, which can further increase the sensitivity of your assay. And over the last couple of decades, there is a plethora of methods being developed for achieving all these different steps. And the currently major used method is the silica-based purification using guanidinium salt and ethanol. The big advantage of that method is it yields very highly pure DNA or RNA, but it also comes with some limitations. The most important ones are the work steps that you have to conduct to achieve this purified nucleic acid in the end. So it's many work steps. That's why it also takes usually quite some time to proceed or perform the whole extraction process. And last but not least, you also have to deal with hazardous substances like these chronotropic salt solutions, the guanidinium salts, and also with flammable liquids like the ethanol, which can make your life a little bit harder in the lab, but also it imposes some major implications if you think about shipping all these products across the globe, because, for example, airlines are not happy about shipping corrosive salts like this guanidinium or flammables like the ethanol. And that is why during the last 1 or 2 decades, it is more and more getting obvious that there's a certain trend towards less purification in molecular diagnostics, and with that towards fast extraction of nucleic acids. And this is basically driven by 3 different points. One is the need. So if you go more towards the, let's say, point of care in diagnostics, and you don't conduct your diagnosis in a central laboratory, then the patient usually gets the results more rapidly. Side effect is, if you think about near patient testing, that a physician who is conducting or able to conduct such a diagnostic gets more profit than compared to the sending the test off to a laboratory and then getting the result back. And more important also in the setting here today is that in some areas you don't have a central lab, and that is actually the thing that you don't need if you go towards the point of care. And that is particularly important in emerging countries, where you maybe have a few very well-equipped institutions in a country, but if you go more towards the communal level, the labs are much less well-equipped. So second driver is the technology itself. If you look at the market, you find so many portable instruments for isothermal amplification, but also for real-time PCR. There is definitely on the assay side so many options to get your diagnostics out of a core or a central lab towards closer to the patient, and with that out of a established lab infrastructure. And the third driver is basically the application itself. All the chemistries that has been developed for the assays more and more get more and more robust against inhibitors. So that means you don't have to have a super highly pure DNA or RNA to basically run a successful detection assay with that. And with all that, it's obvious that the purity can be lower. You don't have to take so much effort to come up with a super 100% pure nucleic acid liquid. And these remaining impurities usually don't compromise the diagnostic results. So, and then you can ask, okay, why should the molecular diagnostics be conducted in the field? Beyond the things I just showed in the previous slide, if you think about neglected tropical diseases, they are usually endemic in mostly rural areas of low and middle income countries, which means in such settings you often lack a very sophisticated or proper lab infrastructure, which makes it difficult to conduct, for example, the full-blown silica-based extraction using either a centrifugation-based column purification, or you cannot also think about big automation instruments doing high throughput extractions and detections. And that is why you should look for something simpler. And also, if you go more towards the point of care, towards the patient, that is actually enabling quite some advantages if you think about screening or detection campaigns. For example, if you go out into a field, you screen a population of a village, and you extract, detect on site, then you can immediately start with a treatment if necessary for the patient or for the person. And the second side effect is that you avoid a possible sample degradation by long transport times, which can sometimes take days or even weeks in some settings, because you immediately after the sampling, you can within minutes go and extract and so save your nucleic acids from the sample that you have taken, and then you can directly do the diagnostic assay. That means for those settings, you have to have a simplified nucleic acid extraction. And if you recall this scheme which I just showed about the different steps of the extraction process, then you will clearly see that the first two, the lysis and the nuclease inactivation, you cannot compromise on it. It's really needed for getting some, to get the analyte out of your sample. But what you can compromise on or change things is the purification itself and also sometimes the concentration. And what is done here, and there are several products on the market doing this, is basically changing this paradigm from nucleic acid purification into inhibitor removal. And the next few slides show you what we at Xpedite have come up with, what solution we have developed for that. So it's this SwiftX technology, which essentially provides a simple workflow, which you can also easily use at a point of care or in a point of care setting. It's basically a three-step procedure. The first step is a cell capture. This is also a differentiator to other fast extraction methods. A cell capture where you can actually use magnetic beads that come with a kit, a binding buffer, and then you mix this with your liquid sample, which can be Can be whole blood, can be urine, can also be wastewater. So basically all sorts of liquids can be applied to this methodology. And in this first step, we first capture the cells and remove the whole matrix, the whole specimen, including all the inhibitors that are in this specimen. And the cells are then separated using a simple magnet and attached to the side of the vessel. And in a second step, these captured cells are then resuspended in a lysis buffer and usually subjected to a heat extraction step, which then performs the lysis of the cells. And simultaneously, the magnetic beads also bind impurities. So that is this kind of reverse purification, because we remove only inhibitors, not capture and purify nucleic acids with that. And the last step is after the cell lysis, that you just put back your vessel in a magnetic rack, wait a minute, and during the magnetic separation, also the inhibitors and cell debris is removed from the lysate. And what you come up with at the end is a kind of cleared lysate, which can directly be subjected to your analytic assay. Like I said, a PCR assay, RPA assay, or even nanopore sequencing is possible with that. So with the SwiftX kits, we basically provide the user with 3 options in terms of the protocol to be followed. It depends on the sample type that needs to be extracted, which protocol is the right one. So if you process solid samples like tissue, biopsies or swabs or concentrated cells, then you can even skip this cell capture step, which I just showed. You only perform this lysis step, so mixing your sample with a lysis buffer magnetic beads, subjecting it to heat extraction, and then put it back into a magnetic rack for lysate clearance. If you work with liquid samples, as I mentioned, like urine or water or other saliva, then you would have an upfront kind of module to this lysis part. You would do the cell capturing and then go with the captured cells into the lysis module. And if you work with more complex liquids, such as whole blood samples, then we recommend doing 1 wash step with the capture buffer before you go from capturing into the lysis module. So right now, or currently, we have 2 different products on the market which make use of this SwiftX technology. One is the SwiftX DNA, and the other one is SwiftX Parabact. And if you look about the functionalities of the 2 different kits, the SwiftX DNA performs the lysis and nuclease inactivation through a heat-assisted extraction using a detergent-containing buffer, but which is formulated so that it can be directly subjected to the assay itself. And with the SwiftX Parabact, we changed this into a different chemistry for the lysis and nuclease inactivation that is basically alkaline assisted, also using heat extraction, and it is showing even higher lysis efficiency for especially gram-positive bacteria. And the nucleic acid kind of purification, which is, as I mentioned, not a real purification, but it is an inhibitor removal. That's why we call it reverse purification. That is in both products utilizing our magnetic bead-based or magnetic bead technology for removal of cell debris and proteins and also carbohydrates. And I mentioned it before, this main differentiator to other products of competitors is this cell capturing that we can perform upfront of the actual lysis step. With that, we can up concentrate tiny amounts of bacteria or parasite cells, such as Schistosoma X, before we actually go into the lysis and purification scheme. So to summarize this, the main advantages why these kits are so useful for point of care settings or other kind of field settings is you don't need a centrifuge, you don't have to deal with hazardous chemicals, you have a strong cell lysis which even can lyse helminth eggs, for example, and you have a very rapid reverse purification, and so the whole workflow is very short, or comparatively short, and also easy to perform. So meanwhile, we have seen quite some uptake of these kits in the schistosomiasis area. There are a couple of papers published over the last few years by different groups showing that the kit can be applied for specimens such as cervico-vaginal lavage or self-collected swabs or urine. And we have also ongoing and future collaborations with also Bonnie's group and Lisette and Erik from Leiden University Medical Center, where we look into extraction of schistosoma DNA from stool samples. And also there's another interesting maybe future application for this technology where we look into enriching CAA antigen of schistosoma from urines for the purpose of increasing the sensitivity of these CAA lateral flow assays. And the few slides I showed you were basically talking about the extraction chemistry, but all this is still useless if you cannot really perform the whole process in a proper way, and if you don't have a proper kind of laboratory environment where you can perform this extraction as well as diagnosis or testing. And there's a so-called lab in a suitcase, which was developed and first published by Ahmed Abdelwahed and Manfred Weidmann in 2015 during the Ebola outbreak in West Africa. And there were also many other publications published later on, which showed this lab in a suitcase. And we have taken this and optimized it in a way that we try to maximize the customization options and also maximize the usability of the whole laboratory. And the next 3 slides, or this and the next 2 slides, show you what we came up with. And so you can imagine it really as a suitcase. It's one suitcase feasible for air transportation, so it is conformed to any IATA requirements. It's an airtight and very robust case, and it is filled with all the instruments and consumables that you need in a field setting. So you have all the instruments, you have all the consumables like the pipettes, and you have also can set up the work table so that you can really work like in a real lab environment. And this is shown here in the next slide. If you look, if the suitcase is opened and set up, then you have an additional LED light for better lighting of the work table. There is the inner surface of the lid of the suitcase is magnetic. And all these holders that you see there are also equipped with magnets, so you can freely arrange the back of the lid with your accessories such as pipette holders, timer, glove box, wipe holders, and so on. And it also comes with mini spinning centrifuge, a vortex heating block, set of pipettes, tube rack holders, magnetic racks. So basically everything that you need for conducting your extraction work. And we also have space for storing samples or reagents in a styrofoam box, so you can even cool it for a certain time. Then there is a space for a detection instrument. That means this whole suitcase lab cannot only be used for the extraction part, but also if you want and need for both extraction and detection. And that's also an interesting feature. The actual work table is a stainless steel plate, which is also slidable, so that you can have access to all the consumables if needed while you are working. And also due to the stainless steel surface, it's also easy to decontaminate. And in terms of power supply, as we know, in resource-limited settings, you often face issues with the electricity supply, and that's why our lab is also equipped with a rechargeable battery, which gives you the option to either work completely independent of any power grids, or maybe just for buffering of the power supply in terms of in case of power outages. And also we supply it, if wanted, with a solar panel, which can be used to recharge the battery while operating the whole lab, which of course then extends the duration of the power supply. So with that, I'm at the end of my more technical presentation. So maybe just as a send off, so why you should choose SwiftX technology. I try to show you that it's very easy to handle, to use the kits, and that's why it's also easy to teach local healthcare workers, even without very deep technical background, to perform simple nucleic acid extraction work. And with that, you would enable them to perform molecular diagnostics on site. We have the cell concentration step without the centrifugation, which, as Bonnie showed, works also with Schistosoma X very nicely. In general, the technology is not only applicable to schistosoma or other helminths, but also to really any kind of infectious agent, from bacteria, fungi, other protozoa, up to helminths, up to viruses, sorry. And last but not least, it's also cost efficient, the reagents, and due to the fact that you need less instrumentation, you also have less investment in for purchasing such instruments. And with that, I thank you for your attention and hand back to Marianne.
Thank you very much, Andy. I think you've really persuaded us there to very seriously consider this SwiftX technology. Thank you also for this really hugely useful overview of the detail of molecular diagnostics, the technology itself, but also what we need to think about really when bridging the gap between what was maybe once considered expensive or complicated in terms of diagnostics and the endemic areas where they're needed. So your vision at Xpedite Diagnostics on how to make molecular diagnostics a reality in the field is really impressive, and so thank you for that. We already had a question from Onyinye Okpai, who's asking, please, how do I get the kits? So perhaps in a few moments when we go into our discussion, you could go a bit more into detail about the next steps in terms of really using and adopting your lab in a suitcase. But before that, we're just going to hear a little bit more from Michael about precisely what does this look like, this lab in a field, all importantly, once you are really in the regions, in the areas where it is needed. So Michael, over to you to give us a little insight on your findings.
Hello, can you hear me?
Yep, very well.
Great. All right, thank you. And thank everybody for the opportunity to talk to you. And great talk from the earlier two speakers. So basically what I'm going to do is to take what has already been shared by Bonnie and Andy, and how we are implementing this on the field in an area with low prevalence of schistosomiasis. So I'll be talking about the evaluation of this real-time recombinase polymerase amplification assay for detection of schistosomiasis in this resource-limited setting. I'll begin by saying that Bonnie expressly, showed us how diagnostic is so important. And just to let us know how important it is, I will just take this quote from the NTD roadmap, which says that effective diagnostics is critical to accelerate process towards elimination, reducing morbidity, and also reducing the program costs. Many a times when I give this presentation, the question is, don't schistosomiasis has already the tools for diagnostics? But just to take us a little back into the same document, the NTD roadmap, which is kind of our guiding principle towards the vision of the 2030, to look at the diagnostic priorities specifically for schistosomiasis. We see that clearly there are tools that exist, but the document clearly shows that these tools still require some major modifications for us to reach the 2030 targets that has been set. And these priority areas are developing or introducing this sensitive point of care diagnostics that we could use in various prevalence settings and also for detection of various species of schistosomes. So this is where my talk will basically focus on. Just by way of introduction, so our question was, can we use these tools that has been developed by Bonnie, that has been designed by Andy, to really detect schistosomiasis on the field? They've really told us how well they've designed these tools in the lab and how well they work effectively in the lab. And we've actually seen some preliminary data on the field. But we wanted to see in actual sense how the system work right on the field with the patient at the communities where these patients are. So we took the suitcase system that has been developed, as Andy said, by Ahmed, and then looked at the assured criteria. The assay has to be sensitive. It has to be able to provide rapid results. It has to be easy to use. And also the reagent has to be stable if you are taking it to the field. But before we went to the field, we wanted to, not just take the assay to the field and not be sure it will work. So we did some in-house validation just to be sure that the assays that has been previously developed by Bonnie's lab and others truly work. So we looked at the, what we called the analytical sensitivity of these assays in the lab. And clearly it shows, as they have previously publicized, that we could detect up to, 1 copy of the air. And also when we did the probate analysis to look at how much, with 90% confidence, how much of this X or DNA can we detect in the sample, we could show with this assays that we could detect with 90%, 95%, confidence that if we have at least 15X in the sample, we should be able to detect it. Again, we also wanted to be sure, is the assay also specific enough? If we take a urine sample and it has other things, is there going to be cross-reactivity? So we took samples. These are what we call standards that we collected for histosoma, haematobium. We also got the X and cell-free DNA. The same we got for Japonicum, we got for Mansoni, both the cell-free and the X. We got other things that could, or other parasites that could be, inside urine, for example. And interestingly, even though these assays were designed specifically for haematobium, we saw that we could also detect the X of Mansoni and even cell-free DNA of Mansoni with the same assay, which was for us interesting. So we took this field to the field. Now we are confident that, okay, this tool in the lab works. It works in the hands of the developers. It works in our hands in a resource limited setting. What happens when we take it to the field? So on the field, we went to a community with low prevalence, in Ghana, we recruited patients. We set up the assay right there in the community, and then we tried to do our testing. So as you can see, this is our team on the field collecting urine sample after a consent were given, collecting urine samples, and then testing these urine samples with the assays that has been developed to see how sensitive we are to detect histosoma X. Of course, before you be able to detect, you needed to go through the sample preparation by doing the extraction. So we took the tool that has been developed by Andy, the speed extract, now the swift extract. And what we did was, after we've spin down our urine samples to concentrate the samples, we took 20 microliters of this concentrate, added the magnetic beads, added the lysis buffer, put it in our heating device for 10 minutes. And after 10 minutes, put it on a magnetic stand for 2 minutes. Then use the suspension for the detection. Of course, we use the RPA, which already have lyophilized or dried reagents. So then it's very easy to use. So we can easily take, directly the sample, add it to the beads, and then put it in our detection system. So this shows you the detection, the amplification and the detection system once the assay has been prepared. Once we got the extracted DNA, we added it to our reagent, which has the RPA reagent mix, the primers and the probes. Mix it, and then added it into our detection device, and then compare the results. So this is just to show you the results, the accuracy of this assay once we took it to the field. So once we performed the RPA on the field, and then when we came the same sample, we ran it also on a qPCR and then compare the sensitivity. And we were able to show that on the field, the assay had a sensitivity as high as 96%. And it was also highly specific. Again, since for schistosoma, microscopy still remains the gold standard, we compare it with microscopy. Of course, there were some discrepancies with the microscopy detection, which has earlier been highlighted by Bonnie. And so the sensitivity when you compare microscopy to the RPA was a bit low. But when you use a high sensitive system like a molecular device like a real-time PCR, the sensitivity is really good, and it's as high as over 96%. So our key message from this presentation and our take-home message for you is that we have shown that we can successfully implement this point of need test in a low risk setting. It shows that it was highly sensitive and specific for haematobium. Even though analytical sensitivity or specificity also shows that we could use it to detect maybe Mansonite, we didn't, evaluate it in this study. But we think that this molecular test has the potential for surveillance, especially in areas with low sensitivity, low prevalence. And we will look forward to, evaluating it in many more settings to see how good this system stands the test of time. I want to end here by acknowledging all the people that have been involved in this study. So this study was funded by the African Research Network for Neglected Tropical Diseases. And we are very grateful to them for the funding to be able to do this evaluation. Again, we will thank the team, Ahmed's team, that developed the suitcase lab, which worked effectively. And then my team in Ghana that were able to put this assay together and evaluate it on the field. Very much thank you for your patience and for your time. And I look forward to any questions you might have. Thank you.
Well, a huge thank you to you, Michael, and I'm sure the audience will join me into thanking our 3 presenters. That's been an amazing hour. not only beautiful science, but also beautiful public health, whether in terms of diagnosing individuals all the way to surveillance programs and really working towards those elimination goals. So a lot of food for thought and a lot of incredibly encouraging work. So thank you, everybody. Lots of comments came through. I don't know if you've had a chance to look on the chat, thanking you, Bonnie and Andy and now Michael for these great presentations. So an all-round big thank you. And I suppose one of the first questions, having really piqued our interest here, what can anyone who's on the call today or any colleagues around the world working in this field, what can they do if they want to adopt some of the technology or some of the resources? What would be their next step? Or what sort of, conversely, what sort of partnerships would you love to establish in order to really deploy the solutions you've mentioned today? We could go in order of speakers. Perhaps Bonnie, you first.
Oh, sorry, sorry, you can hear me now.
Yeah,
good. So it's a hard journey, I think, and we're at this point now where I think a lot of people will want to start to use this. So I think it's mainly first getting in touch with people that have used the technology, and I think about having a conversation about where they're planning to implement the technology, and then to just talk through some of the things that they need to consider. And I've just had a recent conversation with a group in Germany that want to implement this on an island in Lake Victoria, and we just started that communication about what they need to think about. I think in terms of getting hold of some of the kits and things like that, we can invite. We know where to purchase them from. Andy will be the person to talk about how to get those portable suitcases. So I think it's about conversation, really, the first step.
Brilliant, thank you. Okay, so Andy, I'm assuming that you're completely open to all inquiries, and we would encourage, of course, anyone tuning in to get in touch with you.
Yeah, absolutely. Just have a look at our website. There you can ask for contact. We are always open for collaborations. I mean, the field of applications is growing and growing, and we are always keen to also look into new applications which we don't know yet, or let's say where the kit hasn't applied yet. We are always keen to know whether this is also working there, and if it's actually not working right from the spot, then we also especially keen make it work. Yeah, so that's also a big difference to maybe other companies who just deploy kits and then yeah, there you go. But our philosophy is actually very different from that. We really try to support people and the researchers working in the field with our solutions.
Super, and that philosophy really shines through, I think, a lot of the material and projects you presented today. And of course, that partnership slide in all your presentations was really impressive. Michael, how about yourself? Are there particular partnerships or collaborations you'd like to see a lot more of with regards to the lab in a suitcase?
Yes, definitely. We are hoping. Currently I'm in the US, but my studies and everything works in Ghana. I have a team there in Ghana. We are open to collaborate. We already have the system in place in Ghana. We have the suitcase platform already in a research limited setting in Ghana. And the good thing is that we actually were able to put the suitcase together ourselves back home in Ghana. We could also get the tools. It's not so you can buy the system that has been already been designed. You can also put the tools, the system together yourself if you want to. So we are open. You can always contact us, and we are happy to share with you any experiences that we have to get this thing running. Thank you.
Fabulous. And just as we speak, Dr. Obiageli Nebe, Dr. Nebe, who is Deputy Director for the National Schistosomiasis NSTH Program in Nigeria, is just exactly saying that Nigeria will be interested in being part of future field testing in other settings, as the test holds great promise in surveillance. So there's one of the many partnerships, I'm sure, to explore. So some of the first questions that came in actually were just wondering and asking about whether the RPA assay can be used to detect Schistosoma japonicum. That was a question from Gunawan, tuning in from Indonesia, and also from Clarissa da Costa in Munich. What are your thoughts on the feasibility of the RPA tool for multiplexing as compared to, for example, classical PCR? So the message is, can we do even more?
Absolutely. So I can start with that. So japonicum, yeah, there is a couple of papers published on japonicum. So there is a primer probe set that has been developed. As Michael showed, there is cross-reactivity with our haematobiums, what we thought was specific with some of those pathogens. The sensitivity is not quite so high. So yes, absolutely. I think it'd just be a bit more validation for japonicum, and then I think it can be used. I think the only thing with japonicum is to think about stool samples again, as I highlighted lots of times. Stool is a difficult one to work with, so there is thought about using either blood or plasma as well for some of these diagnostics, if that's a sample that can be taken readily. I think on the feasibility, I can't remember what was the question again, Marianne? I can't remember.
So extending to Schistosoma japonicum and also for multiplexing as compared to other. Yeah.
So in that study which I showed, the MIDS project, we are one of our core things as well is about multiplexing, not with other pathogens, but what we're trying to multiplex in is an internal control. So an internal control that shows you if you've got false negatives. So that's our first multiplex challenge. But there is also, for the FGS, we're thinking about multiplexing with HPV for diagnosing women with HPV and FGS at the same time as well. So and hopefully we're going to have a PhD on that starting in September. So yes, we're up for the challenge.
Fabulous. We will watch this space. Just a bit of a detailed question here from Steffi Knopp for you, Andy. My question is, if this bead extraction can be done in high throughput with a multi-channel magnet pipette, or how does the magnetic rack work?
Yeah, so the magnetic rack that we also have is basically working with just standard Eppendorf tubes. But the chemistry can also be applied in a high throughput setting. We have data, it's actually from Bonnie's group or collaborators, right? That this SwiftX DNA extraction was performed using a Kingfisher flex system in really high throughput. And you can basically adapt it to any open source, or not open source, but open standard pipetting instrument, as long as it has a heating source and pipetting opportunity, and also magnetic magnets for separating the beads.
Super, thank you. Another slightly technical question from Yusuf Hamaway. What would be your thoughts on the feasibility and utility of using serum, plasma, or blood samples?
For the SwiftX kits, I guess?
We could start by that, yeah.
Yeah, so yeah, it's applicable to those samples as well. It always boils down basically to what analyte, what pathogen you want to look for. Because if you work with serum or whole blood, then this first cell capture step is very important to get rid of all these inhibitors in those samples. And if the cell capture doesn't work with the pathogen you are working with, then of course the whole method is not applicable to this particular case.
Okay, thank you. Thinking a bit more broadly about applicability, adoption of the technology, there were a couple questions, of course, about cost and cost effectiveness. So a question from Dr. Nebe would be, how do you see the cost evolving compared to other gold standards of points of care diagnostics? And also a question here from Ogechukwu Aribodor. what is the cost effectiveness of this assay and kit? How can it be made readily available to countries? And I think it will be good to replicate this in other endemic countries. So yes, how do the costs compare? How can we make access easier? And also maybe not just thinking about the immediate cost of the technology, but in terms of training users, basically, seems to be a very cost-effective solution. But what hurdles might you see?
So shall I start, and then I might hand over to Michael, because I think the cost is a question that comes up all the time. Costs will come down as more is used, so that is the way that business models always work. I think for NTDs, there is no, it's always going to be non-for-profit, but it's also, we need to keep that cost as low as possible. For a platform like RPA, when you think about costs, it's not just the RPA assay itself, which we've costed about $3.5, but you have to think about everything else that goes around that. It's the plastics, the transport of the reagents, the actual, all those things add up, and that's with any diagnostic. Comparing to others, we have to do that. I think the diagnostics that's coming through for our kind of area in terms of intensity of infection and prevalence is the CAA test, and again it will be that evaluation. How much is that going to cost as well? So that work needs to be done. And I think just think about development pathways for diagnostics. As with the CAA test, it has to go through clinical trial and all those different kind of hurdles as well, to actually make it fully a tool that's actually can be wide, can be provided in a way. But again, yes, we can train and we can provide knowledge exchange, and I think Michael can say about how training his team out in Ghana and how that went.
Yeah. Yes. I mean, I would say also with cost. So, for example, in Ghana, qPCR is costed around $20 per test, right? Mm-hmm. And when we did the cost for RPA with all the extraction kits, especially if you use a kit like the speed extract and all the plastics, the cost is about half of what qPCR will give you. It's about $10. It comes to about $10. So in the context, it's still not cheap, but like Bonnie said, the thing with cost is that it comes with how much you are using it, and over time how we can simplify the system so that we bring the cost down. But also it also depends on the case use of the assay, where you are using it. Are you using it for case and treat, or are you using it for screening? And this is a confirmatory test, so you'll probably be using it not as a screening tool. So these are the terms where to use it. The case use will also affect, how you use the cost. So that is the things that we worked in terms of cost. For training, we have shown that all you need is somebody that is diligent with pipetting. That is all you need, right? So if you get a technician who is okay to pipette, then you are good to go. So basically, that is what you need, on the field. Somebody that can pipette, and then you are good to go. And you can actually train somebody to pipette within a few hours to be able to do that. Yeah.
That's really fabulous. It can't get much simpler than that. So that's wonderful. There was an interesting comment here from Babacar Tiende-Lafaye, and Babacar is tuning in from the IRESSEF, which is an institute in Senegal, the Institute for Health Research, Epidemiological Surveillance and Training. Babacar was just saying it would be great to develop a data management task force. And so perhaps our last question, as I see the allocated time for the webinar is kind of coming up to an end, but we've heard a lot of collaborations. There's a lot of excitement and kind of willingness just in this audience to really use the technology, test it, collaborate. What about the collaborations around the data and the validation and so forth? Would you be open to a task force? Is that already underway?
It's not underway, but yeah, absolutely, we'd be very open to that. And I think what I've really valued seeing over the last few years is other people start to really use this platform. I showed you some of the work in Zambia particularly, and Michael's work to really show you that it does work. I think handing over, just having other people use it and test it and show really good quality data is really important. So I think having this data management and that kind of system would be very valuable.
Andy or Michael, would you like to add anything to that?
Yeah, so from my side, of course, we are always open to support, but to be honest, I don't know what we can contribute as a company to that particular data surveillance, data aggregation question.
Yes, for me, I would say we are open to support. You can always contact us for collaboration. Normally, the limiting factor is funding to get this thing, out there. But once we are able to, secure funding to get this done, we are always able to provide the support for training, especially if you want to use the system in a low research setting. We have the experience in using the system, so we are open to, train and collaborate to get this done within your setting also, or within the communities where you work.
Fantastic. There were still a few more outstanding questions, but I think it's really clear from your presentations and just your philosophy when approaching these issues and this problem that you're very open to collaboration, to knowledge exchange. And so anyone on the call who might still have a question or perhaps thinks of something subsequently, I'm sure there would be absolutely an open invitation to contact the speakers directly. We also can't wait to have you back on ISNTD Connect very soon to hear about any updates and where this is going to go in future. Although it's very difficult to get a date from Bonnie, but I can see why now, because you're so busy. This project has been phenomenal. It's really unbelievable. It's been a very humbling session, I think, and really thank you for taking your time to share all this tremendous work with us. And I'm sure the audience will join us to say a very big thank you. And if there are any final words that you'd like to say before we kind of round off the call to an end, this would be the moment. Perhaps we could go round in terms of the speakers in order. Bonnie, your concluding thoughts?
No, it's great. Thank you very much. All the comments have been actually really great, and certain things to think about as well. I'd just like to really highlight the partnerships, particularly here, I think, with the endemic setting, but also industry. Andy is very much a friend now rather than just somebody I work with. And a company like that Andy has created, they are so open to working with you rather than just providing a product. And I think that's really key. So that's my final comments.
So then it's up to me, huh? Wasn't prepared for any final words, but yeah, thanks for the audience for listening. Also, thank you, Bonnie and Michael, for the collaborations that we had. And as you said, the longer the collaboration lasts, the more you also become friends. And I mean, it's really, really nice to have long-term collaborations with people, and also following together a common path towards a common goal. That's a really nice thing. And just in terms of the questions, I think there were a few addressing the SwiftX technology and also the suitcase lab. So those people can reach out to us, visit our homepage. They can contact us, and we will be happy to answer all these questions.
Thank you.
Yeah, I also want to say thank you to the organizers and to all the attendees for hearing us out, and also to the speakers, to Bonnie and to Andy. You know, we've picked up from what you've developed. We've tried it on the field. It's working for us. We are happy, and we are also open to, you know, others that, you know, want to follow this experience by trying to, you know, utilize these tools in their system where they work. And we are open for collaboration. You can always contact us. I'm sure the organizers will make our contacts available to the audience. You can always contact us. And for me, I'm happy, and I believe for the other speakers to always talk and move forward. Thank you very much.
Fantastic. Well, a really final, very big thank you to you three. Also to the audience, don't hesitate to really delve into those published papers because they are a mine of really lots of information in there. Big thank you to the Global Schistosomiasis Alliance, finally, as co-organizers of this webinar. And we hope to see you all very, very soon, either for the follow-up on this webinar or as early as next week at our next ISNTD event, which will be a 2-day ISNTD festival of all things creative around neglected tropical diseases, as well as an ISNTD Connect on drug discovery for Chagas disease and human African trypanosomiasis. So we look forward to seeing you very soon. And again, all the best to our speakers, and thanks again for sharing all this with us. And we'll see you next time.
Thank you.
Thank you, everybody.
Bye-bye.
Everyone.